Hello students, welcome to another class of biology by Dr. Shabana Khan, and this is another lecture on the series on inheritance, on the chapter on inheritance, and this lecture would be about important genetic terms, right? As we know, inheritance has a lot to do with genes and all, so the first term we're going to look at will be genes. So let's have a look. So talk about genes and what is a gene. A gene is a length of DNA, you can say a segment of DNA, which will code for. So this word comes in again and again. The code is, that means it has a secret code. Code for a polypeptide. All right, and you can also say the word protein, but that's enough. Now, in any cell, there will be 2 copies of every gene. And what are these termed as? They are termed as alleles. How do I define an allele? Alleles are what we say variants of a gene. All right, now alleles can be dominant and recessive. So this word was coined by Mendel. And dominant and recessive, the dominant one means that this is going to dominate the other one, and this will be obvious in the phenotype. So dominant would mean in any population this would be more common, and the recessive one would mean this is rare or this is less common seen in any population, right? Now, we can look at another term, but there are many terms which we can do. Let's first of all have a look at genotypes. Now, genotypes come, this word means, it has a different meaning, but if you try to understand, we always write alleles together, right? So because we have a concept that there will be these homologous chromosomes present in the cell, and the genes present on them together when we write down, for example, capital T and capital T. So these are the 2 alleles and we write them together, that becomes our genotype. But the exact definition of a genotype is that all, you can say, the genetic makeup of an organism. Another meaning could be all the genes and their variants present in a cell. So all of these would be there. So genotype would be all the genes and the alleles present in an organism, but how are they obviously seen? How are they expressed? How do we physically see them? That brings in the second word, and that will be our phenotype. The word phenotype means, it's easy to remember, if like PH in the beginning here and PH in the beginning here, if you want to say F and F, physical. Phenotype is the physical form of an allele or a gene, right? So the word is genotype and then comes the word phenotype. Now when we write them together, like when we write down the alleles together, right? Now, what are the possible combinations that could be there, right? So let's first of all have a look at this. Now, if I have capital T, and if there's another next one allele next to it is also a dominant one, what do I say this is? Or if there is the recessive allele and the allele next to it is also recessive allele, then how do I describe it in words? So the word that is going to represent this condition is known as homo. Homo means same, and zygous. Zygous means a combination or a pair, right? So homozygous could be what? Homozygous could be dominant or recessive. Now homozygous dominant would be this condition, and homozygous recessive would be this condition, and that would mean that both the alleles are identical. So I'm just going to write down the word here. Now, sometimes, you know, in the papers, these definitions are also asked, okay? So homozygous, recessive, homozygous, dominant mean that both the alleles present on the homologous pairs are going to be identical. Either capital T or small t and small t. Capital T means the dominant one and small t means the recessive one, right? Now, a third Condition after that could be what? What if both of them are different? One is a capital T and the other one is a small t. So we say the word we use is heterozygous, right? We don't say dominant recessive, we just say heterozygous, because the word heterozygous itself means it's a different thing. The word other comes from the word heterozygous. So heterozygous would mean that both the alleles are not identical, but they are going to be different. One is dominant and the other one is going to be recessive. Now I'm going to do the definition of a dominant allele again and a recessive allele again. All right. Now what is a dominant allele? Now if we understand, for example, the height of a plant is tall and it is dominant, so this would be there. Now the genotype could be this or this. Now this is the capital T is representing a tall allele. Now if it is present in this condition, the plant is going to be tall, right? So how do I explain this in words? So dominant allele is one that will always be expressed, be observed in the phenotype, or it is always expressed, okay, whether it is in what? Whether it is in the homozygous dominant state or whether it is in the heterozygous state. All right, so you can add this, always will be expressed in the, observed in the phenotype, whether it is in the homozygous dominant state, and that is both alleles are going to be capital, or it is in the what we say homozygous dominant, and this is a heterozygous state. Whether it is in the heterozygous state, that would mean that it is capital T and small t. All right, so again, what's a dominant allele? Dominant, we knew this is one in the population. This is going to be more commonly seen in the population. Otherwise, by looking at the genotype, we can say that this will always be expressed. Whether it has a capital T next to it or a small t next to it, it will always be observed in the phenotype, right? So against this, what is a recessive allele? A recessive allele is one that will only be observed when it is a small t and a small t. So what do you mean? This is termed as homozygous recessive state. So this will only be, a recessive allele is one which will only be observed in the phenotype when it is going to be in the homozygous recessive traits, and that means that the allele next to be also is going to be recessive. If there's a dominant allele next to it, then it will not be observed in the phenotype, only when it is going to have the small t and small t together. All right, now the next one is the word cross. Now this is very frequently used in inheritance, like we say the word genetic crosses. The word cross actually stands for, you might say reproduction, sexual reproduction, or another word could be breeding between two organisms, and another word could be like fertilization, right? So what do you mean by that? We mean that we say when plant A is crossing or is being bred with another organism, which is going to be B, could be two plants, could be two animals, even humans. So this is another organism is crossed with me, that this is going to reproduce with or breed with another, and we want to see the results in the offsprings, right? Now crosses could be like monohybrid cross. You'll hear this word. Also, monohybrid cross means we're trying to study the inheritance of one trait only. All right, and we want to see the probability if we take parents, and then you want to see in the offsprings how does that trait get transferred on. Another one is dihybrid crosses. Di means two, hybrid means a cross. And so dihybrid cross would mean the inheritance of two traits. Now, this is not in O levels, but this will be coming in the syllabus in A2. In A levels biology, you have these crosses. There are tri-hybrid crosses as well, where we take 3 traits and we study the probability of them being passed on to the next generation, right? Now, whenever we say crosses, we like to mention the plants as parents, the plants or the organisms as parents. Parents, you might say, is the first organisms and that we want to study, and another higher term parents are one which are taken as being of the pure breed. Pure breed means that both the genes, both the alleles will have to be identical in them. All right, that's one thing. Now, when parents are crossed, then we get what are known as offsprings. Now, offsprings can also be written down as filial 1 and filial 2. The word filial also means a son or a daughter or a children. So, filial, we can say filial 1 would be the first generation after the parent generation, and filial 2 would be the second generation. And what do you mean by that? Well, for example, if this is parents and then they cross, you get F1, and then when F1 is crossed with F1, we get F2. So these are 3 generations. For example, grandfather, the son, and the grandson, right? So there are 1, 2, and 3 generations, right? Now, moving on, 14. Now, in part of a genetic cross, right? Genetic cross are the flow diagrams chart that we make to show the probability, is what is termed as the Punnett square. Now, Punnett was a scientist, was working in genetics, and he devised a way of putting in the information in a square or a table form, in which what do you have? You have columns and rows. Now, the gametes or the alleles of one parent and the other parent are placed above the column or above next to the rows, and these would be the gametes or the alleles of one parent, and generally the male parent here, if we're looking at the gender, and this could be, so this is like this and this is like this. So these will be the gametes of the other parent of one parent. And whatever the alleles are, for example, I have them here, then the chart, the table is filled up accordingly. This is placed here and then all the way here, and the same goes for this and all the way here, and these are placed in the same way. So you fill up the table, all right, and what do you get now? In each box, you will get the respective genotype, and you place the phenotype in the same box. And all of these things is a certain format of writing genetic crosses that they will carry marks. For example, if this is like 1, capital T and small t, that is the genotype, right, of that organism, of that offspring. Now, what will be the phenotype? Because the capital T is there, so this will be standing for tallness, so this will be tall. Is that okay? And that is for the terms. Our next lecture will be carrying on with the other genetic crosses. Thank you.